Into meiosis, another type of cell division we talked about in class today. This is the type of cell division that results in the production of gametes. Okay, so these are the sex cells. This is how they divide so that you can have a haploid number of chromosomes. That way you can have an organism that has 46 from the combination of a sperm and egg. So anyway, it's 2 rounds of division in meiosis. The original cell has one large and one small type of chromosome. Each of them has already copied itself. And meiosis essentially reduces chromosome number and creates genetic diversity. I'll refer to how it creates genetic diversity as we move forward. Okay, so let's see here. So again, like we said, 2 rounds. We're going to call them meiosis 1 and meiosis 2, and they each have 4 phases. It's going to remind you of mitosis. So you still have prophase, metaphase, anaphase, telophase, but you're just going to have 2 rounds of each. In the first round, you're going to get the homologous pair, which we just briefly talked about today, but the name did come up. You're going to get this pair. Let's say these are homologous chromosomes. They are going to separate from each other. All right, so they're hanging out together while they're going to be separated. Okay, again, they're homologous, meaning they're similar but not identical. They've tried to indicate this on this slide by showing this one in purple, this one in green, but the shape is the same, very similar in both. So they are 2 varieties of the same thing, if you want to look at it that way. Okay, and then there's going to be another round where the sister chromatids are going to divide. So in other words, then we're going to split these 2 apart from each other because we got to get down to that haploid number. And then sister chromatids are copies of the same chromosome. So in other words, this is a it's kind of hard to describe, but this is a chromatid. Oops. And then on the other side, move that back up, is this is a chromatid, and this is another chromatid over here, and these are actually copies of each other. Okay. All right. So we typically take you through the phases like this. You can't see this very well, I realize, so we'll just talk a little bit about the stages, and I'm going to show you a little animation. All right. So meiosis 1, that is the beginning of meiosis. This is going to happen after DNA has been replicated. We know about DNA replication. And as we said, it divides homologous chromosomes in 4 phases. All right, prophase, metaphase, anaphase, telophase, the PMAT that you're used to from mitosis. All right, we'll go into detail on that in a second. Meiosis 2 is going to divide the sister chromatids. DNA is not replicated between the 2. And again, prophase, it's called prophase 2, metaphase, metaphase 2, anaphase is called anaphase 2, and then telophase 2. But notice your result is 4 cells that you get out of it. All right, so let's take a look at, let's do a little animation. Let's check out this animation real quick. This will help. All right, so shrink that down a little bit. Get that in the screen. All right, so we have what's called a germ cell here, and it is too big, so give me a second here. I wouldn't plan on this. Phooey. Come on, get in there. Huh. Okay. How about this? This wouldn't be too bad. All right. So here we have a germ cell. Sister chromatids, as we mentioned before. And we've got our nuclear membrane. We have homologous chromosomes. That is, this green one here is homologous to this purple one. And then we have the other structures of a cell that you're used to, such as the centrosome and centrioles that are involved in spindle fiber creation or formation. All right, so here's how the whole thing goes down. So in prophase 1, The spindle fibers kind of get themselves organized, and then the nuclear membrane starts to break down. Metaphase 1, this looks a lot like mitosis, metaphase, middle. So the chromosomes line up along the middle or the equator. Then this is kind of cool. I'm going to pull this over so you can see the corner here. These are all the different options of what could happen in this next phase are being shown up here. I'll play it again so you can see. But the way these chromosomes line up at the equator would never be the same. I mean, the chances of it being the same every time or ever is extremely small. This is where diversity enters into the equation in regards to cell division. So when a mom and dad have a baby, that baby is a half mom, half dad, but it's a shuffling of all the genes. This is one of the ways in which the genes get shuffled. It's through this stage here with the chromosomes. All right, so I'm going to play this again for you. Try to follow the chromosomes on each side. So right here you've got the two bigger ones, slightly smaller, really small. Two greens on this side, two purples on this side. So watch how it's different throughout each time. All right, so that's what we call random assortment. So there's some randomness added in. So just depending on which chromosomes landed on which side is going to determine which chromosomes wind up in the cells that are created by at the end of this phase. All right, so and also keep in mind, how many chromosomes do we have in human cells? And there's 23 pairs. Here, we're just showing 1, 2, 3, 4 pairs. So just to simplify things, in reality, there'd be 23 across the middle. So a lot more complicated. All right, moving on. Let's check out anaphase 1. All right, let me move that up here for you. Try that one more time. Wow. All right, anaphase, just pulling them apart like the old times in mitosis. And telophase, you get the two separate cells out of it. Okay, so far so good, but now we need to divide again so that we can get down to that haploid number. All right, so this is meiosis 2, prophase 2, kind of similar to prophase 1. You get the nuclear membrane gets all broken up. We get some spindle fiber formation going on. Metaphase 2, lining up in the middle, metaphase. Anaphase 2, pulling the sister chromatids apart. And finally, telophase 2. And you get your 4 cells. And each of these is haploid. All right, so if, say, this was a sperm cell, if it were to join up with an egg cell, imaginary egg cell over here, then you'd get 23 from each. Oh, thank you. All right, so that is kind of a little breakdown of meiosis there. Let's kind of compare meiosis and mitosis. Meiosis has 2 cell divisions, as you saw, and mitosis has 1. In mitosis, homologous chromosomes never pair up. Get this out of the way. Meiosis results in haploid cells. Mitosis results in diploid cells. So remember we said that mitosis is essentially cloning. All right, that doesn't look very good. Mitosis is essentially cloning a cell. All right, it's your body's way of cloning cells because you want to preserve the chromosome number. Meiosis is all about getting that haploid amount so that you can get X and Y chromosomes, egg and sperm made. All right, I'll pull this up. I'll kind of block everything for a second, but here's kind of a side-by-side comparison. And this is in your book as well. But just a quick glance, you can just see, all right, from here to here, okay, once these duplicate, you're going to have a similar, this cell should just basically be a copy of this cell. In fact, both of these cells are copies of this cell. In this case, we had some separation and then and a division, but we had a separation of some chromosomes, and then we had another division and some more separation of chromosomes. And so notice the variety as you move from the original cell all the way down to the resulting, what we call daughter cell. All right, you're just left with some remnants of the amount of chromosomes you had up here. Now you still have the same number. Ultimately, you still have the same number of genes, but they have been mixed. Okay. All right. So we'll do some activities in class to help reinforce this. All right. Gamete formation. That's gametogenesis, is the production of gametes, and is different in males and females, obviously, because we're making sperm and we're making eggs. So it's going to differ a little bit. So sperm becomes streamlined and motile. All right, so notice the little tail sticking out here on these guys. So those are mature gametes in males. Sperm primarily contribute DNA to an embryo. So that's really what the sperm are all about. They're bringing their DNA. Eggs, however, as you might imagine, they also contribute DNA. Again, mom gives half, dad gives half, but the egg also provides a little cytoplasm action and some organelles to the resulting embryo. Kind of makes sense. It's called an egg after all. And then during meiosis, the egg gets most of the contents, and the other cells form what are called polar bodies. So just take a look at this. Sperm, basically what we just talked about is sperm production. Okay, so the whole thing we just went through with meiosis, the gametogenesis for sperm looks essentially like it does for the process we just went through. Where things get a little different is for egg production. So check it out. As was mentioned, as you produce your egg, you go through these steps of meiosis. So meiosis 1 gets you to here, meiosis 2 gets you to here, you get one egg. Well, remember let me move this over just a little bit. At the end of sperm production, you had 1, 2, 3, 4 cells. Well, you only get one egg at the end of egg production. What's left over are called polar bodies, and they are not part of the equation in terms of reproduction. I mean, they don't get involved in uniting with sperm cells. So this is what's going to unite with sperm cells. It's just that the egg just basically takes so much of the original cell's contents, as far as organelles, cytoplasm, and all that. It gets devoted to producing one egg, whereas your other material just kind of gets, I guess you'd say, gets discarded, but we call that other material polar bodies. Okay, that gets us through 6.2.